What's up, everybody, and welcome to this lecture on introductory genetics. And we'll get right started here. So we're going to start off today with expanding nucleotide repeats. Now, what are these? Well, they are very prevalent in degenerative disorders. And what happens here is there's a certain sequence of DNA here. So usually the sequence is in groups of 3, so an entire codon worth of information actually ends up repeating continuously in a certain area. And so shown here can be an example of this. There's simply the same bases repeating on and on. Let's say a common example of this is CAG, repeating continuously. CAG, 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 CAG. Now when you think of replicating this, this chromosome here, consider that when polymerase comes through, these strands come apart, and then they begin to reanneal when amino acids are or sorry, not amino acids, rather nucleotide bases or nitrogenous bases rather come in and they bind onto this new sequence. And if you consider that this side here would have, let's say, CAG, 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 when the strands reanneal, this top strand may end up reannealing at a spot that it's not supposed to. And as shown here, this entire thing would end up over here, as shown, but instead it's reannealed closer, and now this loop part is extra information that will stay in this, as shown here, it will stay in this, this sequence here, and now we realize that in the next bit of time, we have a much bigger sequence here, and it's all CAG repeats. And this is how expanding nucleotide repeats occur. Now you may realize that the larger this, let's say, CAG repetitive sequence is, the better chance of ending up with expanding nucleotide repeats. Consider that when these two strands come apart and they begin to reanneal, if there is more CAG base pairs in a long strand, then it's easier for this to reanneal in the wrong spot. And consider it's very easy for it to reanneal in the wrong spot because all these sequences are the same. CAG, CAG, and if you have CAG on this, or the opposite of CAG on this side, which would be GTC, if you have that on this side, it can reanneal to any part back here as a CAG. And this is what ends up happening in these cases, and the effects are exponential because the longer this strand gets, then the more chances and the more frequency you will end up with getting expanded nucleotide repeats. So you can consider that this issue here is passed on generation to generation, and also consider that in subsequent generations that come about, the effects actually get worse. These sequences get bigger and bigger, longer and longer, and the thing is the effects of these expanded nucleotide repeats get worse in subsequent generations, and the case of or the time of onset of getting issues regarding expanded nucleotide repeats becomes earlier in the lifespan of the organism that comes about from the reproduction of an individual with this this case here. So, as I said, it's a very prevalent degenerative issue here, expanding nucleotide repeats, and an increase in number of repeats is equivalent to an increase in severity of disease. and an increase in age of onset. Usually when the number of repeats reaches a certain number, it causes disease. And below this number, the individual is phenotypically normal. When individuals end up in this range of um, in this number of repeat range that can cause disease, we say they're in a reduced penetrance range. And in this range, they have a percent chance of contracting the disease. And the chance of getting the disease at this point is called penetrance. So this is a degenerative disease. What happens is these coding sequences keep getting these expanded nucleotide repeats, and it alters the protein function in the end of the road after translation. And the proteins, rather than doing what they're supposed to be doing, they end up destroying things. And you can consider kind of why they would end up doing this. Consider that if we take a sequence here, let's take part D here. We'll say that the entire sequence coding for a protein comes from here all the way to here. So we have normal codons here, coding for certain amino acids. So normal, 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 and then we end up getting this repeat sequence here, continuous CAGs, let's say, and then normal, normal, normal. And what can happen is that the proteins end up just kind of messed up in this region, but they do have some ability to do what they used to be doing. So let's say in a neurodegenerative disorder, they can still attach to neurons. They still have the ability on these ends. They can still do certain things. However, they are messed up in a certain way, and when they attach, they can do something different than what they were supposed to be doing. And this ends up causing things like Huntington's disease, where neurons are lost and there's reduced brain capabilities for individuals. So as I said, this is a degenerative disease. Everything is normal in development, but then something destroys that state, and it's these expanded nucleotide repeats. This is different from developmental diseases, where organs develop abnormally in the first place. That's a developmental disease. Another thing to keep in mind here, just a little vocabulary term to know, is genetic anticipation. More repeats in successive generations, as I talked about, leads to more repeats in offspring and increased severity and earlier onset, as I said before. Now here's just an illustration to drive it home how this slippage occurs, where we end up getting a larger repeated sequence. So simply strand comes apart, slippage here, comes back on in the wrong location, and we end up with a longer sequence in this scenario here. So now here we're going to move on to compensatory mutations. These are mutations that let's give an example here. Mutations that, for example, surpass antibiotic resistance, cause the organism to be less fit. So compensatory mutations are just mutations that occur in a normal life of something. And let's say it's some kind of cell that's running into an antibiotic resistance. The compensatory mutation is simply where the cell gains a mutation that allows it to move into this antibiotic resistant region. So it's now, or into this region with the antibiotic, and the cell is now able to, the cell is now considered antibiotic resistant. Now the thing is with this mutation is that it often causes the organism to be less fit. In many cases, another mutation is then presented that causes the organism to return to its original fitness. Now, when I first heard of this, I was like, what? Like, how can this possibly be the case? You gain a mutation, and it allows you to live in certain circumstances better. It improves your overall fitness because you can live in certain circumstances, but it does render your other aspects of your fitness. And then all of a sudden, once you get past the thing that you need to, that is resisting you, all of a sudden you get another mutation that brings you to a state where you were before, and you're perfectly normal and fit. Now this is kind of a confusing situation, but it has shown to be true in experiments. And now, to build on this, it is not a reverse mutation that restores the phenotype, because of a simple substitution or such, to bring it back to this normal point. So this is considered a reverse compensatory mutation. If simply, let's say there's a codon, 3 bases, and one base changes to get another amino acid to bind when or another tRNA to bind and bring in another amino acid that's different. Now, if the single base pair that changes back, that would be a reverse compensatory mutation, where the genotype is the same as the phenotype in before the mutation occurs and after the mutation occurs. And then we'll move on here to look at intragenic suppressor mutations. And these may restore codons, but they will be in a different position. So the amino acid that was mutated in the first place will not be the same one that is mutated back to the original genotype. Can also, it may restore the reading frame via insertion or deletion, or it may restore protein structure via base substitution. Now moving on here to intergenic suppressor mutations, which is simply a mutation in a different gene from the initial mutation, where intragenic was a mutation within the same gene. Different location as the initial mutation, but in the same gene. So, with regard to intergenic suppressor mutations, these are mutations that can occur far from the gene of interest, and these can be mutations on things such as a suppressor or an enhancer of this gene sequence. And when you mutate the suppressor or enhancer, you can increase transcription of this specific gene, and this can have a compensatory effect on the organism's health after the mutation to get past this type of resistance has occurred. Now, interestingly enough, there are some modern approaches to medicine that are actually taking advantage of altering tRNA codons, allowing them to be more easily or less easily, allowing them to more easily or less easily bind when translating, resulting in altered protein. Some of these can recognize early stop codons and do many other things. So simply changing the tRNA codon can have very unexpected, interesting effects. As I said before, stop codons, they can say there is a certain stop codon and we change the tRNA codon and we distort one base pair, or one base on the codon to affect the wobble position of a part of the gene or the mRNA sequence, and simply changing this one base may stop the tRNA from binding. And let's say it's a stop codon, it'll stop it'll stop from adding this amino acid at the stop codon site, and therefore translation rather will not stop. And this is a great way to do many things of value for individuals in the medical field. So now moving on here to mutation rate. And this is a little interesting thing to note. So mutation rate is mutation frequency minus repair multiplied by probability of detection. So that's the mutation rate formula, you can say. And some organisms may not want a low mutation rate. That's something to think about. And why would this be? Well, for example, viruses survive off their ability to mutate quickly and get through resistance. So as shown here, viroids up here, they have a small genome size and they have a very high mutation rate. And this, of course, as I said, is very beneficial for viruses. They live, or they don't live, but they're not alive, but they're successful on their ability to mutate very quickly and get into places and get through resistance that may be preventing them. So moving on here to transposable elements. And so first off, I'm going to name two types of transposable elements that are found in humans. These are DNA transposable elements, as shown on the left here, and also retrotransposons, as shown on the right. And so DNA transposons are simply, they simply have a target sequence, and this target sequence is, it has terminal inverted repeats. As you can see, it goes A, B, C, D. And there is an enzyme called transposase which can recognize this certain sequence, cut, and allow a transposable element to be inserted in. Usually the cut is actually sticky. It is cut in kind of a formation like so, like I'm showing on the screen here. And this allows polymerase to come in and put in the bases to make sure that this transposable element is perfectly fit into this location and it's integrated completely. So this transposable element is moving via cut and paste. So it comes out of a certain sequence and then it gets pasted into a different location as shown here. So these transposable elements are genes that are capable of changing location in the genome. Well, almost about half of the human genome is capable of transposition, and being capable of this transposition is dependent on terminal inverted repeats, as I said here. The transposase is able to recognize this sequence and cut it to allow transposable elements to be inserted in. And same here in the case on the right, as shown, is retrotransposon, how it works. Not all transposable elements here contain transposase. As I said, transposase is capable of cleaving, making staggered cuts, and allowing transposable elements to be inserted in this area. But they don't all have transposase coded for them in this element region. Many don't, and they're still capable of moving because they can just use other transposase found in the cell. As long as the transposases make a cut, these transposable elements are still capable of moving even if they don't code for this transposase anymore. And in most cases, these sequences would originally code for transposase, but through mutation and after time, simply their ability to do this may be lost. So looking at retrotransposons here, they move via copy and paste. So you have a retrotransposon, it copies, as shown here, into RNA, And then an enzyme called reverse transcriptase takes this RNA and codes DNA from it. It makes 2 strands of DNA, and then it inserts itself back into the genome. And they're very likely to be simply mutated sequences of viral DNA that have been inserted into genomes before, and they use this reverse transcriptase to move, which is what viruses actually use. Each time it takes place, there is another retrotransposon. So as you can see, this continues over time. As this continues to occur, you end up with more retrotransposons in the genome. And you can probably think over millions of years, now most humans are at the point where they have a whole lot of retrotransposons that are taking up space and not doing much. Well, they may be doing something, but we're not at the point yet of understanding this stuff to say what exactly they do. And at the point we're at now, they have not much of a function to what we can think. So one popular study of transposable elements was in corn. And as shown here is some corn. Interestingly, each kernel is actually a unique individual. And when a transposable element is found in a pigment, this is just how these corn kernels work. When a transposable element is found in a pigment, the gene color is the color of the gene is white, or not the gene, sorry, but the color of the kernel is white. And when there is no transposable element in the pigment gene, the color is purple. Now it's hard to see in this black and white kind of image, but there are instances of some purple, and this would be yellow. This guy over here would be entirely purple, maybe spots of yellow definitely in this case here. And so there are many possibilities of color that it can occur with these transposable elements. And if the transposable element got into a pigment gene in meiosis, the offspring would be white, and if it did not get in meiosis, the offspring would be purple. Now, if the element got into the pigment gene in mitosis, patterns of white could occur, and if the transposable element left in mitosis, purple patches would occur. So, as shown here, there are many options that have come about for these corn kernels. So I'm just simply saying that transposable elements, as shown in the previous slide, can get into a certain gene, and if they end up in a certain gene, then in this case, a pigment gene, which gives rise to the certain colors in these kernels, if the transposable elements end up in this gene during, let's say, mitosis, since that's in somatic cells, a few of the color of kernels will change, because when the transposable element is in the pigment gene, it turns the kernel white. And of course, if the transposable element comes out during mitosis, then cells would turn purple. Some of them would. Not entirely all of them, because it's mitosis and it's just a simple cell. All the daughter cells of the cell will be a certain color here, like let's say in this case purple. If the transposable element comes out of the pigment gene in mitosis from one cell, its offspring will become purple. And so you can see here just a small part of this entire kernel here is purple because it occurred in late mitosis. Now, if it occurred in meiosis and not in mitosis, that the transposable element either inserted itself in that pigment region or left that pigment region, then a kernel would be entirely white or entirely purple, depending on which case. So now moving on here to transposable elements, the consequences that come about from these transpositions. So one is insertional mutagenesis. It can interrupt a gene, as shown, let's say in the pigment in the pigments of corn kernels. It changed the normal purple color to white. It can also cause early deletions, just as it inserts into another gene. It can distort a gene sequence by changing the sites of exons and introns. It may move other genes with it around as it does this transposition, and it could cause non-allelic homologous recombination. And as we know from previous lectures, this gives rise to duplications and deletions, which may cause adverse effects for offspring of individuals. And also, other things that it can cause is it can introduce a new transcription start site. It could introduce a transcription factor binding site. It could introduce a new stop codon, and it could move heterochromatization. And in that case, we know that that would be altering the chromatization of a chromosome, and therefore rendering that genetic sequence unavailable to any type of expression. And I hope you guys learned something, and that's the end of this lecture. I'll see you next time.